Hello everyone, welcome to join TERMIS Pre-Accelerator program. This is Tracy Yu, Chair of the TERMIS EU BPC Committee. Today we're gonna have the webinar session 2, and it will be about building an evidence base. Through the webinar, we will have Dr. Nihal Engin Vrana give a keynote lecture, and then Dr. Maxi Dragovits give the second talk. Then we will answer the question at the Q&A session. Please remember you can submit questions throughout the session. You can see a question window on the control panel, so you can submit questions at any time during the webinar. We will all answer the questions at the Q&A session. Now we will have our committee member, Olive Bo, give a recap of the PAP session 1.
Hi everyone. So just to recap very quickly what we went over in October for the first seminar, we had a really great introduction to the regenerative medicine industry from two speakers, Dr. Chris Giannetti, who's at Harvard, and introduced by Dr. Engin Nihal Vrana at PROTiP Medical. PROTiP are developing some really great artificial larynx devices, which hopefully we'll hear a bit more about later. And then we heard from Dr. Giannetti a little bit about the state of the industry at the moment, starting off talking about some of the innovation that's really driving the industry at the moment, particularly around the design of our therapeutic products and cells, but also around the manufacturing processes that are involved with producing these products. Second, moving on to the financing of these things, and that covered a little bit about investment, but also about reimbursement, which is particularly interesting for gene therapies, where some innovative financial models are coming to the table here. And we moved on to looking at some of the clinical success stories that we've had in the industry, predominantly around the CAR-T field, which I'm sure many of you have heard about. There's been some very, very excellent clinical data come through. And as a result of that, we've seen an awful lot of competition around those platforms. But inversely, we've had a few regenerative medicine and tissue engineering products which haven't met their clinical endpoints, and we've got quite a large range of efficacy and safety data coming out of the industry at the moment. So then we looked at a little bit of the case studies of commercial successes that have been achieved to date, and Chris's comments were really that there haven't actually been many commercial successes, if any, so far. And he started to talk a little bit about how this might change over the coming years. And then he's finished off talking about the regulatory hurdles. And you know, there's an awful lot of regulatory frameworks in place regulating term products. And navigating those is something that companies have had to really grapple with in a heavy way over the last few years. And Chris was quite optimistic that most of those challenges have been addressed now, and that in the future we're going to be pretty good at addressing the regulatory barriers, following the examples of some of the successes that he showcased. So let me close up with some interesting questions for the panel, which hopefully some of you who attended last time took something away from, and we'll have a similar kind of setup in the coming seminar. So I hope you look forward to it.
So thank you much, Oliver, for the recap. And same as Oliver, I would just like to highlight that you can ask Q&A questions throughout the webinar, so please do, and we will answer your questions during the Q&A session at the end of the webinar. So it is my pleasure to welcome back today's keynote speaker, Dr. Nihal Engin Vrana, to this second webinar for our PAP series. Dr. Vrana is the Vice President of PROTiP Medical, a French company active in the field of otorhinolaryngology. Beyond his work as Vice President, he also currently coordinates an EU Framework Programme 7 project, Immodgel, and has a strong interest in the valorization of the foreground of multinational research projects with small and medium enterprise SME partners. So thank you very much, Engin, and please begin when you are ready.
Thank you very much for the introduction, and welcome to everybody. So today, Mercedes and I will talk about building an evidence, and we have a strong belief in multinational projects. So that is the main backbone of the talk. I will try to show you what can be achieved with the multinational projects and try to give you some concrete examples, and then Mercedes will talk about how you can actually build such projects and what you should do to obtain funding for such a project. So as in the last presentation, I would like to start with our ongoing project, which is called Immodgel. It is a collaborative multinational EU project, and it is about immunomodulation around implants. But it is a structure based on biomaterials, bioactive agents such as growth factors and cytokines, and autologous cells, specifically autologous macrophages. So by definition, it is an advanced therapeutic medicinal product, and it needs to follow the necessary regulation for that. So if you have a tissue engineering or regenerative medicine product, it will fall in this classification too. So this is why we are going to start with this. And in this project, what we are trying to achieve is that by specific control of macrophage phenotype, control around the micro environment of the implant, so that we can control the immune response to any implantable device. And for this, when we have this idea and when we have the consortium, we had a development stage. And in the development stage, the kind of evidence that is necessary for you is the basic research to better understand your system. And in our case, this was a polarization of the macrophages and its relevance to implants. The second thing, if you have a biomaterial component, you need to optimize its structure and what is its relevance to the final effect of your system. So for our case, this was a hydrogel system, so we needed to go through the optimization of hydrogel and show that it has the effect we are after. And also in the case of an implantable device, there is also other relevant physicochemical factors that you can leverage to get the final therapeutic result you want, such as the topography of your system, the stiffness of your system. And within the context of the project, these are the things we looked into. And finally, in most cases in tissue engineering and regenerative medicine, you will have a bioactive component, which is such as growth factors like BMP or VEGF. It can be cytokines, it can be an amino acid, the polypeptide, but generally this is a necessary component, and in our case we try to optimize the concentration and the composition of such components. So this is the development of your system, and within a project the next step to look is the testing. And to test your actual final product, you should have the right testing environment for the proposed action for yourself. And preferably, you should have this first in an in vitro model, such as an in vitro elaboration of the structure you have. Here is one example. What we are claiming is that we will affect the cells that are in contact with the implant through these macrophages we have. So we try to develop a model of such interactions in vivo by having two different hydrogels. One is a model of soft tissue, the other one is our Immodgel system, to see how they interact over a course of time, which we think relevant to the in vivo events. So you need to have structures like this. And through this validation steps, you will get an iteration of the development of your design. You need to improve your design to fix unforeseen problems, to go back, make several iterations, and finally, you need to show a proof of concept in vivo that what you propose works in the way you proposed. So this is how we developed the project, and we are now at the level of development of in vitro models and in vivo testing. So this is the basis. Now what I'm going to show to you is the successful examples of this process, which has gone all the way to the end. The first one is actually the world's first stem cell-based therapy approved in the world, and it is for people who are blind due to burns, particularly due to the lack of epithelium, corneal epithelium. So this is a product called Holoclar, and what they do is they expand human corneal epithelial cells which contain a stem cell subpopulation ex vivo from the patients, and then reimplant them to cornea to provide the patients with a healthy, renewable corneal epithelium, so that provide a means for patient to see. So this obtained clearance in EU in 2015. And for this, I mean, as you are developing your product, this is your endpoint too. And you will be authorized only when you can show the safety, efficacy, and quality of your structure. And there's a specific committee looking after ATMPs, which is called CAT, Committee for Advanced Therapies, and this is within European Medicines Agency. So it will be a very good source for you to look into this, and you can actually ask questions to this committee. They are quite open to questions because this is a field, many things are ambiguous. So as you are developing your product, it is a very good idea to be in contact with them. For the specific case of Holocry, here you can see two main publications coming from this. One is from, as you can see, from 1997. This was the very first clinical demonstration. This was done with 2 patients, showing that they can restore vision by their method. And the second one in the New England, coming from in 2010. This is the actual clinical proof. Made with more than 100 patients, showing that it is efficient, it is stable for a long time, there are no side effects. So this is the route that you need to go through to achieve a product that is authorized. It is not for scaring you actually, but this took 25 years. But you should, the way I see this is that these people are the pioneers and they opened the way for us. So it took 25 years for the first product, but that doesn't necessarily mean that from now on it will be like that. Because of all the experience developed in this previous cases, we will be able to do things faster. I gave a link to a Nature article, interview with the leading person in this project, Professor Graziella Pellegrini, which I was able to work in a European project years ago, 10 years ago. This technology was in development, and it was partially involved in the project. That's how I know this technology. And I suggest you to read that too. She gives you a very good idea of how things developed over time. So as a postdoc, she came up with the concept in 90s, most likely the same case with most of the people who will be in the competition. And then from 90 to 97, they optimized their protocol and they applied it in people. And then during the optimization, as they are looking after a subpopulation, they need to see whether the subpopulation is there or not to be sure that the efficacy is there. So in 2001, they discovered a marker which helps them to evaluate that, which makes things much easier. And through that, by 2010, the exact number I think was 112. They have treated 112 patients, and by 2015, it is authorized. It is a company now. This protocol is available in several hospitals around Europe, and most probably soon it will be around the world also. So this is the very end point, and I will also give you an example of another success story, which is close to the endpoint, but is still in the development stage. This is a French company called Defimed, and this is a good example of how you need to have certain evidence, convincing evidence, so that people can fund you for further development. Defimed is coming from 2 EU projects actually, and the main product that they are developing is a bioartificial pancreas containing cells for treatment of type 1 diabetes. So again, it's a long story. They started in 1996 with now we're at Horizon 2020. Before that, it was frameworks, and this was a 4th framework. They first did the project in framework 4 in 1996, a 3 or 4 year project. And 2004, they obtained a continuation project, which gave them a prototype of an bioartificial pancreas. So by the end of that project, they have a bioartificial pancreas prototype. And with that, they went to the local agencies and obtained some national and other international grants. In 2010, they were started to be incubated in Strasbourg. And in 2011, they founded the company. They have carried out experiments in primates. And there was a recent newspaper article saying that this year or next year they will start applying their product in humans. So this is again a long study, but again, just like the other case, this is the groundwork. So things will be much faster from now on. So this is the kind of evidence you need to develop. And as you can see, this is a long process. It needs many parts. And because of that, you need collaboration. And because of that, you need the collaboration in a very structured, organized way. And international collaborative projects provide you with this. And also, I want to say something about the concept of evidence base. Like evidence base has its roots in clinical use. It comes from the understanding that anything you propose to patients or do to patients should have a strong evidence base. And since tissue engineering and regenerative medicine mainly at the moment is trying to come up with medical solutions, we should follow the tenets of the evidence-based medicine. And this has three components. The first component is the systematic review of literature. And in this case, it's not just a review of the literature, but a good understanding of your technology and its positioning with respect to the current state of the art. This is very important as a starting point when you are starting to build your evidence base, because you need to base your evidence with other findings too. The second thing is extension of this is meta-analysis of the available literature and available results, and to be able to show where your experimental work stays with respect to the aggregate results in the relevant field. The third part is the randomized control trials for any medical intervention. And in the specific case of tissue engineering and regenerative medicine, it is important to think, because they are generally innovative products and they are not really replacing something existing. It is more like something new. So you really need to think about what kind of scenario is necessary for testing your product or idea clinically and preclinically. And what are the measures that you need to implement to prevent bias? Not just the randomization, but to really show that the effect you are getting is due to what you are doing, not because of other confounding factors, because most of the ATMPs, by definition, the reason they are advanced, they have many components. So you really need to have a good idea of where the effect is coming from. And this requires proof of concept in both in vitro and in vivo in the right testing medium, which means that what you are saying that you are doing should be shown in that exact conditions. Sometimes this is available in some animal models. There are some animal models for some specific conditions, but sometimes that you need to develop this yourself. The other very important concepts are the safety and efficacy of your system. You need to make the risk assessment for your every single component and the possible side effects and the stability of your overall system. And also you need to have a very good understanding of the mechanism of action of what you are proposing and the added value of each component you add to the system, because each additional component means additional expenditure to just show their safety and an additional parameter to understand your system. So everything you add should have a definite added value to the final medical application. And one thing that is very important, and this is another reason why you need to be involved in big projects, is that you need the fundamental research to understand the underlying science of what kind of mechanism you propose. Like if it is a differentiation of cells, you need to have a very good understanding of what makes them differentiate in the way you want and not in any other way. Or if it is about the effect of a certain kind of molecule, you really need to know the pathways that you are dealing with. So it cannot be just from A to B. You need to mark every point as you go along. So I will just give you one very tangible example from our side. We, as Oli said, we have an artificial learning system, and it has been implanted in humans. And this is the overall system. And at the bottom of this system, we have a structure, a porous structure that helps the connection between the trachea and the implant. And in the end, when we have the whole structure ready and integrated through this porous system, we can implement also artificial epiglottis-like structure. So this porous part at the bottom needs to be optimized so that it gets integrated as fast as possible. And it is based on microbeads of titanium. And we started with trying to find the optimal conditions for this. And this was published in 2014. This is another thing when you have a strong scientific data, it is important to show it in scientific publications. So our hypothesis was that we can control the integration of the system by the size of the beads, and there's a direct relation between the size of the beads and the behavior of connective tissue cells on these structures. So we have started with different bead sizes and try to see first in vitro how cells interact with these structures, if there's a difference between the population of these structures with respect to the size of the beads. And our initial observations were that with the smaller beads, we have much more deposition of extracellular matrix molecules, and cells can make bridges between the beads much easier compared to the bigger beads where they have the tendency to stay on the beads. And based on this in vitro evidence, we went to the rats and tried to see whether the integration of these implants will be different with respect to the size of the beads. And within 3 weeks, we can see that when we have the big beads, we have empty areas in the thickness, in the cross section, whereas the bead size, as the bead size goes lower, we have better integration of the structures. So with respect to these information and together with the available safety information, we were able to decide on a optimal bead size that we use in clinic. So this is one example of how build the evidence for one part of your structure. You need to do this for every component of your structure. So with that, I thank you for your attention.
So our next speaker is Mercedes Dragovits, who did a PhD at the University of Heidelberg in Germany. She is project coordinator for Immodgel, which we will be hearing about in a moment. This is a collaborative research consortium of 8 different partners across academia, industry, as well as technology transfer stakeholders, and they're very much focused on the kinds of concerns that we're looking to address in this seminar in generating an evidence base that your technology platform works. The Immodgel platform, which I won't go into huge amounts of detail on because I suspect we might hear about that, is a sort of a device that aims to reduce the risk of rejection following implantation of devices or tissues. And Mercedes has a particular focus on intellectual property, which I think would be a really interesting thing to get some questions about. So if you do have some questions about anything that either Nihal or Mercedes are talking about, please put them into the chat box on the right-hand side on the control panel, and hopefully we'll stimulate some really interesting discussion at the Q&A session at the end. So without further ado, I will pass over to Mercedes, who will give us a bit of a talk about the focus of our seminar today. Thank you, Mercedes.
Thank you very much for the introduction. So yes, I was just told I'm Mercedes Dragovits. Just to introduce myself a bit, I'm a biologist. I studied in Germany, and now I'm working since 2013 at the Steinbeis Europazentrum. This company is a technology and knowledge transfer center, and it's part of the Steinbeis network. It's Steinbeis Foundation in Germany. It was founded 26 years ago under the mandate of the Officer of the Commissioner for Europe of the Ministry of Economic Affairs in the region of Baden-Württemberg, which is in the southwest part of Germany. We have 2 offices located in Stuttgart, which is the capital city of Baden-Württemberg, and in Karlsruhe, where I work myself. Our aims are linking enterprises and different organizations, research organizations, with innovative partners all over Europe. And we initiate and accompany European cooperations on research and technology. And for this, we are involved in several projects. And these projects are mainly funded by the European Commission. And I'm presenting a few steps in general, how you should proceed. to get EU funding. First, you should identify a suitable call from the European Commission, like a framework program, as Engin mentioned before. The last one was FP7, the Framework Program 7, and now the EC has launched Horizon 2020, which ends in 2020. Then you should develop on your project concept and check that your project idea is aiming at all the aims of the European Commission as stated in this call. You should form a consortium accordingly so that all the aims are being reached in a good quality by the partners. And in the last step of the project planning, you assign the different tasks to the consortium partners, you assign task durations and budget to the different partners. So for the consortium formation, we use the Enterprise Europe network, which is also an EU-funded project. It's a network consisting of over 600 partner organizations, not only within Europe, but also all over the world. And around 3,000 people are working in this network. The aims of this network is to assist organizations in the different regions where the partners are located in finding European cooperation partners and in identifying funding possibilities. We also help with an aim of internationalization. We assist SMEs, so small and medium enterprises, with innovative products and services. We support the collaboration and the networking between SMEs, but also with research institutions. We are directly connected to the European Commission, and we assist in finding European cooperation partners. So if you're interested in looking at the different services that the Enterprise Europe network offers, you can go to the website here on the slide, and you can look for a local contact point who will be able to assist you. So here I show the track to EU funding. I first talked a bit about the identification of a call. And once you have formed your consortium and have a clear project idea, you submit your proposal. So now I'm focusing more on the Immodgel project. Here I'm working with Engin together. We are coordinators of this project. And the first step was submitting the first stage proposal. So this was a two-stage submission model. The first proposal, the short proposal, was submitted by the end of 2012. 2 months later, it was approved for the second stage, which was submitted in February 2013. A few months later, it was approved for EU funding, and after a short negotiation phase with the European Commission, we were able to kick off the project in October 2013. So the consortium of Immodgel consists of 8 partners, as already mentioned. We are located in 6 different partners. The first one is the administrative coordinator, the Steinmetz Europazentrum. We have 4 universities, the University in Strasbourg, in Heidelberg, Nottingham, and one specialty of this European cooperation project is that we also have a partner located outside of the European Union, namely in the United States. This is the Brigham and Women's Hospital with affiliations to Harvard and MIT. And we also have 3 industrial partners here. The scientific coordinator, who's based in Strasbourg too, at PROTiP. We have an Estonian SME, ProtoBios, and a Czech SME, ContiPro. So the types of organizations in this project is, well, the first one is the Technology Transfer Center. We have 4 research organizations and 3 SMEs. The role of the administrative coordinator, which is, in this case, it's me, is on one side the administrative, legal, and financial support to the partners, the management of know-how within the consortium. We coordinate the dissemination activities of the partners. We also manage IPR rights, so intellectual property rights, and exploitation of the project. So we are creating an exploitation plan for this project. It is really important to have one partner who focuses on this kind of activities, because it helps having a smooth and effective collaboration within the consortium. We make sure that the deliverables and milestones are accomplished in time as set out in the proposal. We also encourage our partners to reach a high quality of project results and also the reports. And in case there were any problems and conflicts, we would be the partner that mediates to prevent them or solve them in case they should arise. And we keep an eye on the project budget and expenditures to make sure that the partners are administering their budgets according to what was set out in the proposal. The academic participation in such research projects is, well, you can imagine that it's important to have generation of scientific knowledge. As Engin already mentioned, it's important to publish your results. Also, for the dissemination of the project and to increase its visibility, it's important to participate actively at national and international events or conferences or symposia and congresses. We have 2 universities that are linked to clinics and hospitals, and this enables the access to patient samples, which in such projects is also vital. And through that, we also have access to surgeons who are in the end are end users of the end product of the project. As for the industrial participation in research projects, the European Commission is aiming at having commercializable outputs of such projects. So they're interested in having commercial products that can reach the market after the project ends, or if manageable during the project duration already. It's also vital to have an IPR strategy. So we are regularly searching patents and to make sure that we are not infringing any patent in case we want to file patents. For the moment in the Immodgel project, 1 patent was submitted this year. But this whole idea of who is going to patent what, this was already planned before the project was launched in 2013. This is part of the project planning. You need to define who is going to own which IP, who is bringing which background to the project, and who is going to generate which kind of IP. And one really important thing is to take care that everybody is aware of this patenting strategy within the consortium, because if in case you were to publish something you want to patent, before you patent it, you are blocking yourself from patenting. So you should wait until the patent has been filed before you publish. So one of the other things that makes industrial participation vital in such projects is that there is a market orientation in the project, and the partners they have an intrinsic motivation for marketability. And the project outcomes of this Immodgel project, they will be exploited also beyond the EU funding period. The EU funding period goes from October 2013 until September next year. But since we are generating these patents, we are going to create revenues through licensing, and we're also thinking of setting up spin-off projects, probably also funded by the European Commission. And also the idea is to create a startup based on these patents that are being generated. So I wanted to show you what the Immodgel system is about. So as Engin mentioned before, he uses titanium implants, and we have partners working on an adhesive layer based on a polyelectrolyte multilayer, which will glue, so to speak, the titanium surface to the hydrogel, to the immunomodulatory hydrogel containing cytokines that will control the phenotype of autologous macrophages that are also encapsulated within this gel. the concept of the project will be as following. First, we will take blood samples from patients. The cells will be isolated, and in the first step, these cells will be diagnosed. They will be analyzed in terms of the patient's immunoprofiling. We have 2 partners working on this immunoprofiling, and this is supposed to generate another patent. And on the other hand, we have a foreign body response in a chip to analyze what the reaction of the specific patient is to specific implant materials. The outcomes of these 2 different analysis tools will be fed into a database, and according to the results of this analysis, we can decide which kind of material we're going to use and which mixture of cytokines should be included in the hydrogel so that the macrophages don't promote an inflammatory reaction but rather inhibit it. So once these parts are designed and developed, they will be adhered to the implant surface with this adhesive layer, and this will lead to the final Immodgel system that will lead to improved implantation outcomes. So if you're interested in the dissemination activities of Immodgel, you can check our website. You'll find it under www.immodgel.org, and you can check our publications, press releases, events the partners are participating in, and also brief profiles of the partners involved in the project. We issue every 6 months an electronic newsletter to reach not only through the scientific publications, we reach rather the scientific communities, but through the electronic newsletter, we also have the possibility to disseminate them through each partner's network, so we can reach other types of audience, like stakeholders. And in each newsletter, we focus on one specific topic that the project focuses on, and we also present one specific partner in detail. So far, we've had 5 releases, and 3 more are upcoming. On the tab for scientific publications, you will find a large list of publications that were generated since the project started. So for the moment, we have already 26, and more are in progress or in review process. So until the end, as the project ends, we expect to have more or less a total of 40 publications. Some of them are open access, and you can download them directly from our website. We also have a social media presence, mainly on Twitter. So if you are interested in following us and finding out about highlighted event participation or new scientific publications, you can follow us. And one final slide on the final conference. We will be hosting a symposium on immunomodulation and tissue engineering and regenerative medicine at TERMIS in Davos in Switzerland next year. And also the PROTiP, the coordinator, the scientific coordinator of the project, is sponsoring a business plan competition. So if you're interested in knowing more about our project, you can either visit us there. So that would be it from my side. Thank you very much. And I pass again to the organizers.
So thank you very much for the very interesting talk, Mercedes. And I think we are now move on to the Q&A session. Our audience has submitted some very interesting questions. So the first one is to Mercedes, and it is about what exploitation involves. You mentioned exploitation in your talk, and does it refer to the commercial outlook? And is it about how Immodgel project aims to go forward following the closure of the research project?
Yes, exactly. That's the idea. I've mentioned the patents that are being generated, and based on those patents, the idea is to create a spin-off company I don't know if you want to add something on that.
Yeah. I mean, in the sense, the exploitation is that there is a view in the beginning of any kind of project. The idea comes from a certain base, but as you go along, you discover other possible applications or other possible exploitations, and a TTO office helps you in finding the right application to have the right strategy to be able to exploit your system. So that's the main help you will get from a TTO in the sense of exploitation.
All right, thank you very much. So the second question was, should a researcher make a decision on setting up a spin-out company or not based on only preclinical findings?
My personal answer to this, I will give a personal answer and I try to give a more objective answer. My personal answer is that if the researcher believes that what he has in his hand is something he would like to work for the next 5-10 years, yes. It is a question of conviction about the final output of what you are going to do. In the end, it might not work out like that, but it is worth doing it. So that is the personal answer. The objective answer is that the nature of the preclinical findings. Are the preclinical findings are relevant enough for the final application so that you can really commit yourself to it. If you are someone more defensive, you might want to see input from the clinicians about to have opinions about different stakeholders to see whether where you stand is strong enough or there is more research needs to be done before coming to a commercial stage. So there it is more about the quality of your results, the preclinical results. But from a personal point of view, for any given project, if you believe that you have something in hand that can go as far as you imagine it to go, then it is always good to follow that kind of dream.
All right, thank you very much. That was very interesting. So the third question is, how do you think IP strategy is developing within cell gene therapies? Are direct claims of a product composition necessary?
The answer to that from my part is that if the composition is essential to the activity of the product, then yes. If changes in composition have only incremental effect on the final activity of the product, then no, because then that is not an essential part. But like in the framework of tissue engineering, if you have a very specific scaffold or if you have a very specific bioactive agent that changes the whole activity, then there is something patentable and it is worth patenting. The main difference I feel in this context, in the context of tissue engineering, is that these kind of patents are not really licensable in general sense because they really need additional development. So you need to think about most of the cases you are the one who is going to valorize this patent. So do you want to keep this information to yourself up until you go to the end of your research that you have something that is exploitable, or you want to have a patent to see whether you will find a partner? This is a strategic decision and there is no yes or no to here. It is again depends on the situation. I will say that for a patent on a product composition, the product composition should do something really special. It shouldn't be just something that people inherently intuitively feel that will work. It should have a component that is really making a difference.
All right, thank you very much. And so in your talk, you mentioned that the first product that got from clinical concept to regulatory approval took 25 years. I'm wondering whether you could comment on the timeline that is sort of generally true these days for products. Does it still take maybe 15 years or 10 years? Can you maybe comment on that, please?
I would say it will be between 5 to 10. The reason these kind of products take 25 years are the concepts need to be developed with the product. So we are talking about the stem cells in that specific product, and the understanding of the action of the stem cells, their behavior, has developed during that 25 years too, so that's why it was incremental. Now we have more mature information, so to put up things together is much easier. For example, same case for us, like this polarization of macrophages has been around for, you might say, 10, 15 years, but the real understanding has started to come in the last 10 years. So we have something to base our assumptions on. So at the moment, I would say more to 5 to 10 years, because there's an inherent timeline to this development, because at the end we are trying to heal something, and if you want to show the healing, you need to have enough time to show the healing, enough time to show the efficacy, and enough time to show that the healing is not temporary, that it has something that really increases the life quality of the patient. So those kind of things, there's no way to rush. We haven't had the technology yet. But as for the development part, I think we are luckier compared to those people, because more information is available to us, so we can develop a system much faster.
All right, thank you very much. That was very interesting to hear. And I think the last question for today is, so when we build an evidence-based and want to attract investors, and also at the end we want to get regulatory approval, of course, but should our strategy should it be different for investors versus regulatory approval? For investors, should we show more about the profit and perhaps, and for regulatory approval, of course, more safety and efficacy is important. I'm wondering whether you could you comment on that, please?
What you will talk about between these different stakeholders will change, but the essence won't change, because the investors maybe do not want to know the details of the regulatory process, but they would also like to know that you will be able to go through the regulatory process. And at the same time, for the other type part, is more important to focus only on safety, because that is their job and that's what they care about. But with the investors, I mean, the story doesn't change. It is just the details will change. In one case, you need to show the safety of each of your components and how it is going to be used. What is the benefit compared to the risk? And this you need to also explain to your investors for them to understand what they are putting their money on, but maybe not in the same level of details. And then you can speculate in the sense that make predictions about what kind of market size you will have, what kind of conditions you are going to solve, and when do you expect this to be available for the patients. Those parts investors would be more interested in, but the essence of the information won't change. The level of detail in different parts will change.
Perfect, thank you very much. So I think with that, I'll conclude the Q&A session, and we'll hand it back to Tracy for wrapping up our webinar. So thank you very much again to our presenters.
Thank you, Richard, and thank you, Ollie. I would like to thank again to our fantastic speaker today, Engin and Mercedes, great talk. And also we would like to thank our program partners, Immodgel Project and PROTiP Medical, for their generous support. If you have any more questions you would like to discuss and ask our speakers, please feel free to submit them at TERMIS EU BPC LinkedIn group. You can see the sign here at the bottom of these slides. Also about our session 3, the next session will come in 3 weeks, in mid-December. We will talk about regulation in tissue and regenerative medicine of ATMPs. We will have speakers, Julian Hitchcock and Christopher Bravery. Also, we will have our guest session chair, Magna Roberts. Thank you again to everyone who joined us today. We look forward to seeing you at next session. Thank you.